materials called the neonicotinoids. And you probably know that nicotine is a nerve poison. You may or may not know that if you had a drop of pure nicotine and put it on your finger, very shortly thereafter, you'll be dead. You probably may not know that there's enough nicotine in one cigarette to kill a person. But fortunately, it doesn't all come out in one drop. It comes out as the person smokes, gets into the blood, gets to the liver, and gets denatured. Okay? So, nicotine's extremely toxic. So the chemists at Bayer, or Bayer Aspirin, as you know them, Bayer Crop Sciences, worked with the molecule or an analog of the molecule until they got to the point where the vertebrate nervous system really isn't susceptible to this molecule much at all. But the invertebrates are really, really susceptible to the way they built this molecule. So you can buy the material now to put drops on your dog and cat, and it will go systemic in their bodies. And then when the fleas bite them and eat them, it will kill the fleas. And if they're used in the field, they're pretty darn good at controlling particularly sucking bugs and some of the mites and whatnot. So there are a number of ways that they can be used. You can put it on the seed before you put the seed into the ground. You can put it in as what we call a side dressing, which means that you run down the row and incorporate a little of it off to the side of the plants. And then when you irrigate or when it rains, it gets down into the roots and it'll come up into the plant. There's a root injection, and you saw that word merit before when we talked about eucalyptus. That's the root injection, merit, around the eucalyptus tree. And it can be put on as foliar. And no matter how it gets to the plant, it gets into the vascular system and basically into all the tissues. And this bothers the beekeepers because most of the beekeepers think, well, then it probably gets into the nectar and it probably gets into the pollen. And my guess is they're probably right. Okay? But try to find that data. That's very difficult. So the first blow-up occurred over a material called gaucho, which has imidacloprid, and that's the one they're all after. It was used as a seed treatment on sunflowers. So you would take a sunflower seed, put a little bit of this imidacloprid powder on it, plant the sunflower seed. The sunflower grows, and the imidacloprid gets into the sunflower, and then when it comes into bloom and the bees are visiting, the beekeepers claimed that they got into so much imidacloprid from the nectar in particular, but maybe in the pollen, that they lost their memories, it erased their memories, and they couldn't find their way back to the hive, and their hives went down, like CCD. And the idea was they lost their memory. Well, the beekeepers and the regulatory people fought back and forth, back and forth, back and forth, and finally France, Spanish legislators said, okay, enough, no more gaucho. So they got rid of gaucho. What happened to the bees the next year? They went to the sunflower, and they went down, 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 down. What was the reason? Residual gaucho in the ground, says the beekeepers. Okay. It's been about 5 years now. What do their bees do now when they go to the sunflower? Down, 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 down. I don't think gaucho's necessarily the problem that they thought it was. However, if you take imidacloprid in the lab and check it out, it kind of makes the beekeepers look like, you got a leg to stand on. Because you can take a honeybee and put it in what you and I would call a honeybee straitjacket. All that's sticking out is its head. And you take a little tube and you blow a stream of air onto its antenna. And then you incorporate into that stream of air some kind of an odor. And when the odor hits the antenna, you then touch the mouthparts of the bee with some sugar syrup on a swab. And the bee will stick its mouthparts out and it wants some more. So you feed it a little bit. And then you wait and you hit it again and you feed it a little bit. In about 15 minutes, you can train a honeybee to always stick its mouthparts out when it smells that odor. And the bee will do that for 30 consecutive days without any more reinforcement. And I can tell you now, not the exact part of it, but that's being used by our government. Okay. Now, that's the easy part. What happens when you incorporate a little bit of a neonicotinoid into this procedure? First, let me tell you what happens when you introduce a little bit of nicotine into a human. When I was at the University of Massachusetts as an undergrad, my roommate used to smoke cigarettes. Only during the time he was studying for exams. And I said, what on earth are you doing? And he said, I learn better when I smoke. And I thought, you're nuts. Later, I got me a book from Consumers Union called Licit and Illicit Drugs. And they titrated nicotine into humans through an IV. And sure enough, you give humans just a little bit of nicotine, and it actually stimulates the nervous system, and they can do better work and faster work on math tests. Then you give them a little bit more, so you're beginning to shake up the system a little bit, and they go back to at about the same level that they would have been at if you hadn't given them any. And then if you just put a little bit more in there, well, then you're really beginning to slow them down. And guess what? The neonicotinoids, this one in particular, imidacloprid, works exactly the same way on the honeybee. With a tiny bit of imidacloprid in that swab syrup, they will learn in 5 to 10 minutes what it used to take 15 minutes to learn. And they'll hold that for 30 days. You put in a little bit more, and they go back to learning at the normal rate. And you put in just a little bit more, and we're talking about very, very sublethal doses here. And they don't learn worth beans. And not only that, if they've already learned, and you give them that third level, they won't respond. So again, the beekeeper said, there was the memory. I don't think it's that simple. You have to take a big look at what's going on here. There's an odor coming in and hitting an antennal receptor. And this is right up Walter's alley. And then that's supposed to send some kind of a nerve impulse, through the antennal nerves. And then there's a connection there, and then it moves over here, and it eventually gets into the brain. And then if it's going to be a memory, it moves up into the so-called mushroom bodies. Okay? But then if you're going to have the mouth parts come out, that impulse has to come back down out of the brain, around over where the mouth parts are, and get in there and stimulate the muscles, to act right and all this sort of thing. So how can you say it's the memory? The memory is just the mushroom body. That could happen anywhere. But indeed, it does stop the bees from acting normal. So the beekeepers are again convinced, loss of memory. That wasn't the only experiment. They put up elongated tents. And they put a small colony of bees that went in, and they put a feeder at the other end with sugar syrup in it. And after a few days, you have flight going back and forth like crazy. That's where they're getting their food. So you start putting a little bit of imidacloprid into the syrup. The first thing that happens is the bees fly down, and they don't just land and start drinking like crazy, which is what they do when the syrup is normal. They kind of hesitate a little bit. But they drink it, and they fly back home. So you put a little more in. Well, now the bees are going down, they're not too crazy about it. And so they land on the dish, and they walk along the outside and whatnot. But they finally drink it and go back home. So you put in a little bit more. Well, now the bees almost, they just don't look like they want to do this at all. But they finally get down there, and they drink it. And now they try to get back home, and they got a real problem. They bounce off the sides of the tent. They fell on the ground. They're completely useless at getting back home. The beekeepers would probably tell you that the reason for that is that they lost their memory. But I think the reason they were bouncing off the walls and whatnot is because they were intoxicated. And one of the reasons I say this is because if you pick those bees up off the ground and keep them in the lab under the right conditions so that they don't die, in a day or so, they kind of come back to normal and will begin to fly back and forth to regular syrup again. So their memory's not erased. It's just that they were intoxicated badly. However, if that happened in the field, they wouldn't go home. I agree. Well, as you might guess, Bayer has decided they got to do something about this. This is a pretty black eye for their stuff. We better put out some kind of information. So they did. They put out a fact sheet. And it isn't just a one-pager. It's a little fact booklet. And the important thing to glean from that is the fact that they established that for imidacloprid and a couple others that are very, very similar in their toxicity to honeybees, it probably takes 192 parts per billion if the bee drinks it to kill the bee. Billion. We aren't looking for chemicals at parts per million anymore. We're looking at parts per billion. Okay? Now, apparently, because these papers are published, Baer went out and took a look at what happens when you treat sunflowers and canola with imidacloprid and said, our studies show about 5 parts per billion imidacloprid in the nectar, and so under those circumstances, there's no way it can be hurting the bees. Well, there's a researcher down at UC Riverside who took a look at what was going on out in the field because something was going a little bit wrong in his biocontrol projects. And some of his biocontrol organisms were drinking nectar from the red gums when you could do that, when it's available in the winter. And merit had been injected around the roots of the eucalyptus tree in order to get rid of the lerp psyllid, which is a sucking bug that beats the trees up pretty badly. We've got them out behind bee biology, and our eucalypts look awful, but we'll never do this. And they found approximately 550 parts per billion in the nectar. So that's well above the 192. Eventually here, I'm going to tell you one of the things we need real bad is somebody to study that sort of thing. So beekeepers do want to know how much of that material is getting into the nectar and pollen. And as we mentioned before, France got rid of gaucho, Spain got rid of gaucho, France got rid of fipronil as well. But when you read the scientific documentation, it basically doesn't back up the policies, because there have been times when they've actually taken imidacloprid in concentrations higher than you'd ever get out of the plants, put it into the syrup, and put the syrup feeder right on the colony and left it there for 6 weeks. The bees did fine. They overwintered fine. There was no CCD. They were fine. So as I said, we get to this, we need in the system somewhere a honeybee toxicologist to determine what effects these pesticides are having on the bees. And we used to have 3. 3. Larry Atkins at UC Riverside, Carl Johansson, and Dan Mayer up at Washington State University Pullman. We now have 0 working on this. So I think that's a gaping hole that should be filled.